Okay, welcome back for another lecture in the human species. Today we're going to talk about evolutionary theory. But before we get started, I just wanted to remind everybody that our first exam is 1 week from today, on Tuesday, June 9th. So the exam will just be a multiple choice exam focused on the first 4 lectures. So the 2 previous lectures we've had, this lecture on evolutionary theory, and on Thursday's lecture on species and speciation, along with the assigned readings. So this exam will be available for almost the entire day, from 9 a.m. till 9 p.m. On June 9th. It's not just available for the class time. The reason that I'm doing this is to give people flexibility, because I know people have different schedules, and I'm just trying to be as accommodating as possible. You'll have 2 hours to complete the exam. It'll be timed, and after you finish the exam, or after those 2 hours, it'll submit automatically. So no matter whether you've completed the exam or not, it automatically submits after 120 minutes. As you take the exam, it tells you how much time you have left. So it'll say, like, you've got 15 minutes left, 10 minutes, 5 minutes, etc. The other thing is that because it's available until 9 p.m., it automatically submits at 9. So if you started at 8:59, it'll submit automatically after 60 seconds, right? So you have to start the exam, if you want the full 2 hours, by 7 p.m. So I'll say that again. You have to start the exam by 7 p.m. if you want to take the full 2 hours before it submits. For the exam, you'll have 2 attempts. Should say, just in case you have browser or internet problems. So let's say your Wi-Fi goes out for whatever reason. Your dog steps on your thing. I don't know, right? I'm just giving you 2 attempts just so you have it. Inevitably, somebody will ask for an additional attempt, but I'm not going to do that, right? You have 2 attempts for those reasons. I'm not going to give you a 3rd attempt because your grade wasn't high enough. That's not how this works. So make the most out of what you have. The other thing to note is that this is an exam, so you're not permitted to use any lecture materials, any PowerPoint slides, Google. The kind of questions that I'll ask anyway aren't the kind of questions that could be answered easily. So it's a really bad strategy to try to cheat on the exam. You're not going to have enough time to do that anyway. So just take the exam in good faith. To do well in the exam, you should study the lecture videos that we have every Tuesday and Thursday. You should study your lecture notes that I hope you're taking on the videos, and you should study the assigned readings. There've been a few questions about this, about what from the readings am I going to put on the exam? Of course, that's a question that I cannot answer. What I will say is, I'm not going to ask you anything on an exam that we don't talk about on these videos. So if there's something that we talk about that's in the reading and on the lecture, then you know it's important. If there's some obscure thing in a reading that has nothing to do with what we've talked about, then I'm not going to ask you about it. Okay? But yes, the readings are important, and they will show up on the exam. And so after this lecture, I'm going to post a list of Exam 1 key terms and concepts to sort of guide your studying. It's really important, in my opinion, that when you study for an exam, not just for this class, but for any class, that you do it seriously and you write things down by hand, because that slow, tedious process helps you remember things. For me, if I was taking this course as a college student, I wouldn't be able to remember anything just by listening to these videos, or even by typing on my computer. So when I was an undergraduate years ago, I couldn't take notes on my computer very effectively. I just couldn't remember stuff. So I had to do it by hand, which is slow and boring and kind of old school, but it's the way that works. So if you want to get the best grade possible, make sure to to write stuff out by hand. Okay, so that's the first exam. Um, Please send me an email if you have any questions. Send me specific questions about things that maybe you don't understand that I can help you understand, right? So this is really critical because we're doing this in a remote fashion. It's a little bit more difficult to communicate. And so it's important that on your end, if you're not understanding a concept, that you let me know before the exam. If you tell me after the exam, then I can't help you. So please, feel free to email me if you have questions. Okay. So again, today's lecture is focused on evolutionary theory. And so what is evolution? We've talked a little bit about evolution, but we haven't really dug in deep. We hear about evolution in maybe like a pop science kind of way sometimes. You hear about so-called evolutionary debates and that kind of thing, but like what really is evolution? Well, there's a couple ways that we can think about it. The first is that evolution is a change in gene frequencies within a population over time. So what is an allele? What is a gene? And what is a frequency? Basically, we're just saying that genes are changing in a group of living organisms over time. Remember that an allele, is just a variant form of a gene, right? So if you have genes for eye color, let's say, you have different alleles for different eye colors. And so in a population, if the percentages of those alleles are changing over time, then that's evolution. Another way to think about it is a change in phenotype within a population over time. And that is sometimes referred to as descent with modification. Okay, well, what does descent mean? And what does modification mean? Well, descent just refers to like one species or population coming from another. And modification just means a change. So if populations or species of animals, and that includes humans, or plants over time are changing, that's evolution. So the main question that we're asking in this lecture is, how does evolution occur within populations? How do gene frequencies change? How does the phenotype change within populations over time? Okay, so there are four main forces of evolution. There are four main ways that evolution occurs. Apologies if you can hear background noise. There's somebody vacuuming outside of my apartment door, so that's a thing. Okay, so the first force of evolution is mutation. And again, a force, four forces, right? Another term, it's just a way that evolution happens. Okay, mutation is the first one. And mutations refer to changes in the sequence of A's, T's, C's, and G's in your DNA. It refers to a change in the sequence of nitrogenous bases in your DNA. And so this can happen during cell division, where basically when the sequence of DNA is copied during cell division, mistakes can be made. Think about taking notes and you're transcribing something. You may miss a word. You may leave a word out by accident just because you're trying to keep up with something. You may misspell a word by accident because you're trying to keep up with something. And so the same thing happens in DNA. Sometimes things get left out by accident. Sometimes new things become inserted by accident. Sometimes things are substituted or sort of like misspelled. And so these are the three sort of kinds of mutations. You have a substitution, where you've got one base, in this case a cytosine, that's mutated, it's substituted for a different one. A different one is substituted there. The next form is insertion, where a new nitrogenous base is inserted into the sequence, where one was not there before. And the third is deletion, where something gets left out from the original sequence. And so mutation, as I mentioned, occurs in cell division. It can occur during like meiosis and mitosis. Mutation from an evolutionary perspective is most relevant during meiosis, or the formation of sex cells or gametes, because those gametes basically act as the thing that transmits a parent's DNA to their offspring. So, for example, if a sex cell has a mutation, it potentially can lead to some change in an offspring. And so mutation, along with meiosis and the genetic recombination that occurs during meiosis, is responsible for producing variation within populations. It's the reason why kids aren't just carbon copy sort of combinations of their parents' DNA. It's important to note that some mutations can be neutral. They do nothing. There's no change in the protein that is coded for by that stretch of DNA. It could be bad. It could be deleterious. It could have some negative effect. Or it can be positive. It could be beneficial, right? So mutations are not always a bad thing. Mutations don't always do anything, but it's a typical normal process associated with cell division. I'm forgetting the estimate for the number of mutations in each cell, each new cell, but it's like hundreds of mutations, right? So this is just a normal thing that happens all the time. So that's one way that evolution can occur, mutation. The second force, or the second way that evolution happens, is something called genetic drift. Genetic drift. Genetic drift is just random change in allele frequencies over time. It's a little bit difficult to understand if you don't understand that each offspring and the genetics or the alleles that they inherit from their parents is kind of like a coin flip. And so genetic drift is strongest in small populations. And so here's like a hypothetical example of how alleles or genes can change in their percentage in a population over time. And so here we have two alleles. We have allele A1 and allele A2. And through the force or the process of genetic drift, alleles can be eliminated. They can be lost. They can go to zero frequency in a population, or they can become fixed. 100% of everybody has the allele. So it looks something like this. Over time, you have a normal change in this allele. If you start out at 50%, right, this allele occurs at 50% in this population. Over time, it increases slightly, decreases slightly. It's drifting around this central kind of value until eventually it goes until 100%. It becomes fixated. Here's another allele in the same population at the same period of time. That allele, through random chance, through genetic drift, can become lost. It can decrease to zero. And so you can imagine, like, if you started out with a really big population of organisms of some sort, and for some reason there was a decrease in population size, and now there's only two individuals left, one male individual and one female individual. If they have an offspring, if they produce an offspring, it's likely that that offspring will not inherit one of the alleles that the parents has, just because again each offspring gets one chromosome from mom and one chromosome from dad at each of the 23 pairs, at each of the 23 locations. And so, just by chance, that individual may not inherit one of the alleles that the parent preserves. That allele in that parent that the offspring does not preserve is the only case of that allele existing in this now population size of two individuals. And so if that offspring doesn't inherit that gene or that allele, then guess what? That allele goes to zero in the population. And that's genetic drift, right? So genetic drift is a form of neutral evolution. There's nothing beneficial or bad, deleterious about any of these alleles. They're just changing in their frequency over time within populations and can be affected by drastic population decline. And so this is why like conservation of animal species can be important, because frequently small populations of endangered species don't have the genetic variation to sort of bounce back from disease or other issues. And so that's genetic drift. The third force of evolution is something called gene flow. Now, keep in mind that these two forces both start with G, right? So, like, I know that new terminology, can be confusing, but so just keep that in mind. Try to separate genetic drift and gene flow in your mind. Okay, so gene flow goes like this. Let's say you have two populations, population 1 and population 2. Each of these dots represents an allele or a gene within the population. And you can see that populations 1 and 2 differ in the alleles that they possess. And so if individuals from population 1 and 2 encounter one another somewhere in the world and they produce offspring, well, now genes are flowing from one population to the next, because the parents of these new offspring passed on these new alleles. So sometimes gene flow is referred to as migration with reproduction. I feel like that's a really weird way to explain it, just because migration of like a population over some area in the world is like the mechanism. It's part of the mechanism for how this works, but it isn't the thing itself, right? So gene flow is when a population changes. Let's say population 1 changed in the composition of its genes or its alleles, because individuals from this population inherited those genes from individuals in population 2, which have different genes, right? So genes are flowing from one population to the next through sexual reproduction. That's gene flow. And now this population differs over time. So that's evolution. Gene flow becomes important in a variety of contexts, but it has an interesting role in understanding human evolution, because over the last like decade or so, we've realized that based on the ancient DNA preserved in extinct human species, that gene flow happened, that early modern humans, early members of our species, Homo sapiens, interbred with previous archaic populations of now extinct human species. So we'll mention that in a little bit more detail in a later lecture. All right, so that's gene flow. And these three forces of evolution are very important, but the fourth force of evolution gets a lot of attention. And that force is natural selection, or we could even refer to this as selection, because there are different forms of selection, with the natural, the quote natural one being one of the more important ones. So you may have heard of natural selection. You may have taken a biology course in which natural selection was discussed. And so hopefully some of this is familiar, but if not, that's okay too. Okay. So in under the process of natural selection, or under the force of evolution of natural selection, genes or alleles that improve, quote, fitness become more common. And so what is fitness? Is this like physical fitness? Is this like your ability to run long distances or lift heavy weights? No. We're referring to evolutionary fitness. And fitness refers to basically like the number of healthy offspring you have that survive long enough to have their own offspring. So if you have a lot of healthy kids that survive to have their own kids, then your evolutionary fitness is high. If you have no kids, for whatever reason, then your evolutionary fitness is low. So anything that improves the ability for you to survive and reproduce and have healthy offspring becomes more common in the population, because your kids inherit those things that helped you survive. And so natural selection is like a very intuitive process, right? It's just a sort of emergent property of nature. Okay, so in order for this to work, we need three things to happen, right? We have three necessary conditions. Number one, there has to be variation in the trait, right? In the trait that the alleles code for. If there's no variation, then there's nothing to select, right? So this has therefore nothing to do with selection in that case. So for example, if for some reason really tall people, 7 feet tall, had a much better chance of surviving and reproducing compared to much shorter individuals. I'm not 7 feet tall myself, right? So I would be at an evolutionary deficit. Then you might expect taller individuals to have more kids who inherit the genes that code for being tall, and so their kids would be tall. And so over time, you'd have an increase in tall individuals. But on the other hand, if everybody in the population was the exact same height, then your height is irrelevant for your survival. Or in other words, your height is not something that helps one individual survive and reproduce to a greater degree than another individual. Okay, the second thing is the trait must be heritable, must be shaped by your genetics in some way. We know, of course, over the last couple lectures that this is a complicated component, because even though traits can be shaped by genes, they're also shaped by the environment. And so under natural selection, we're referring to the genetic portion of the variation in a particular trait or a set of traits. If the trait is not heritable, then the surviving parent doesn't pass it on to the kid, right? So the trait has to be ultimately shaped by your genetics. Number three, the variation in that trait must influence your fitness. It must affect your ability to survive and reproduce. Otherwise, all individuals would just pass that stuff on to their kids. So we mentioned that fitness is the probability of producing surviving offspring. The evolutionary sort of game, I suppose, for living things is to increase fitness as much as possible. Usually it's measured as the number of surviving offspring produced. And it's important to know that fitness is measured relative to others, right? So again, like if everybody has the same trait, if everybody has the exact same height or whatever the thing is, even if it's heritable, if it doesn't influence your fitness, or everybody has the same fitness, then the trait is not going to become more common in the population. Okay, so natural selection, we usually think of this, we talked about alleles and genes that affect fitness. We talked about like phenotype in terms of like physical traits. I've been mentioning height. We've used height in previous examples, but it can also influence behavior. And so in this book, The Extended Phenotype by Richard Dawkins, the phenotype is not limited only to biological processes, but extended to include the effects a gene has on its environment. And so it's kind of interesting here where we know that the environment exerts a selection pressure on species. The environment also, through development, uh, influences the expression of the genotype. But species also change their own environment, right? So there's like this feedback loop thing that's happening. Um, and so this can include traits that influence an organism's fitness. And so many behaviors are complex traits, right? They aren't simple Mendelian traits, like we saw with the pea plants, right? It's not like, oh, you have the gene for as a beaver for like building a dam, or you have a gene for building a spider or a spider web as a spider. These are complex traits. And so these are examples of like some of the ways that different organisms can modify their own environment through their behavior. So I mentioned like building a dam, building a spider web, building other structures. We do it as humans in certain ways, but we'll mention that humans are kind of like an extreme expression of a lot of this stuff, and the fact that humans have such enhanced cognition and such complex culture and long lives, it means that culture, like the way that the kind of like shared set of behaviors and rules that we have in different societies around the world, act in some way as kind of like a buffer against some of these natural processes, right? So humans complicate this matter to some degree, right? But here's an example of an animal creating an environment, modifying its environment through the formation of a spider web. And so behavior can evolve. And so here's a really cool example of an experiment that began in the 1950s in Russia, where they started with a population of wild foxes. And the thing that they were wondering is, could we create like domesticated foxes through evolution? And so they started with a population of wild foxes, and they tested their reaction to humans. In this case, their reaction to humans would be considered the phenotype. And some of the foxes were aggressive toward humans, whereas other foxes, and in those cases, they didn't breed those individuals. And so these individuals had lower evolutionary fitness. They didn't pass on their aggression to their offspring. Other foxes reacted to humans in a friendly or cautious manner, and in those cases, they bred those individuals, and those individuals had many offspring. And so fitness for these individuals was increased, fitness for these individuals was decreased. And so in this case, this decision about which individuals bred and which individuals didn't is the selection part, right? But in this case, the scientists were selecting who was breeding and who was not, as opposed to nature. And so in this case, this is considered a form of artificial selection because humans are the ones doing the selection, right? But selection nonetheless. Okay, and so over time, guess what happened? Take a guess. What will happen to the population of wild foxes here after many generations, right? After 50 years. We now have friendly foxes that were domesticated by these people in the 1950s. And so the really cool thing about this is, number one, it shows that in a relatively short period of time, over like 50 or 60 years or whatever, or 70 years, that um, this population can change fairly dramatically. And so now you have these foxes that are much more dog-like, right? They're much friendlier to humans. And so the second cool thing about it is that selection for one trait, right, selecting and breeding individuals who are friendly, led to changes in many traits. And those traits were not the target of selection. They just sort of came along for the ride. And so here are some examples of the traits, right? A star, like a patch on the forehead. You can see it right here. Floppy ears, right? Ears that are kind of floppy to the side. A short tail and a curly tail. And so you can see that in the selected group, these are the friendly foxes, per 100,000 animals, the selected group has more individuals with these traits compared to the wild group. And so selection for friendly foxes led to a series of other changes. And that's due to the effects of something called pleiotropy and integration. And so pleiotropy is the idea that one gene can affect many traits. Because again, these are complex traits that are associated with tens or hundreds or however many genes at the same time. And integration refers to the fact that changes in development can have broad effects. And so basically all of these traits, having a star, floppy ears, different tail morphs, they're all linked to the things that code for friendliness in a fox. And so that's really cool. It's a really interesting example of selection on a short-term scale. When like farmers or breeders like breed certain plants or certain animals to increase certain characteristics of those species, that is selection. Right? The fact that we have dogs and so many different breeds of dogs, right? A dachshund, a golden retriever, Saint Bernard, like all of these different breeds, that's the result of artificial selection, right? That's the result of humans breeding dogs for different purposes. And so dogs, sort of similar to the friendly foxes, first evolved from wolves, right? So like a wild wolf is a close relative of the domestic dog. Okay, so it's important to know that evolution works with what it has, right? Usually traits are pushed or pulled in one direction or another. It's not that common that truly like sort of spontaneously new traits appear. And so this is due to something called homology. There's a few different H words that we'll encounter. This is one of them, homology. And it's similarity between different animals due to the fact that they share a common ancestor. And they inherited their similarities with one another from that same common ancestor that also had those things. And so here's an example of the homology of the limb bones in reptiles, mammals, and birds. And so even though all these different animals use their limbs in different ways, and even though the limbs of these different animals look fairly dramatically different, they're still made of the same parts. So each of these, so we'll use the human as an example, right? There's one upper arm bone. This is called the humerus. You don't have to worry about the names for those. There's two forearm bones. That's the radius and ulna. Then you've got some small bones of the wrist. Those are carpals. And then you've got metacarpals and phalanges of the hand, right? You can see your own metacarpals and phalanges of the hand, right? 5 fingers and 5 toes. And each of those elements is made up of the same number of bones, right? So in the thumb, if you look at the phalanges of the thumb, it's made up of 2 bones. If you look at the foot, the big toe is also made up of 2 bones. Whereas all of the other digits are made up of 3 bones. And so different animals, again, reptiles, mammals, and birds, also have these same constituent parts, these same bones that have just been modified through the evolutionary process. And so bats, for example, even though they have wings, they have a humerus, they have 2 forearm bones, they have carpals, metacarpals, and phalanges. Right? The bat's wing is supported by the bones of their upper limb, by the bones of the human hand. And that's because the animal that is the common ancestor that all of these different animals evolved from also had these bones in some form. And so in other words, each of these different groups has similarity in the bones of the skeleton that's related to the fact that all of these guys evolved from a common ancestor. That's homology. And, you can think of many traits that are similar between different species. Right? The fact that all primates have a thumb and a big toe that act as a grasping organ is due to the fact that primates evolved from a common ancestor who had that thing. And so you can think of this as like traits that run in your family, right? Like, people, my family typically have like this facial characteristic, and we all kind of recognize each other as members of the same family for that similarity. Individuals of that family inherited that from an ancestor, like a great-great-grandparent or something who had that same trait. Right, you see pictures online of, here's my grandmother from, the 1800s and here's me today. They look very similar. They're similar because one of them came from the other, right? The grandchild came from the grandparent. It's the same thing here, but on the scale of a species. All of these species came from an ancestral species that also had these things. So that's homology. There's also similarities in living things that are not due to the fact that they share a more recent common ancestor. And so similar traits or similar, quote, solutions can evolve independently in different living things. And so if you have like 2 different species who look different, but over time they live in similar environments, in different places, with similar selection pressures, with similar things influencing their survival and reproduction. In those cases, we see the evolution of similar traits. And again, we call that convergence or homoplasy. Convergence. They're converging, the 2 different populations are converging on a similar thing. And so this happens over and over again in living things. And so eyes are an amazing, striking example of independent evolution, right? So vertebrates, humans are vertebrates, like things with a vertebral column and skeleton, compared to insects who don't have a skeleton, an internal skeleton like we do. We both have eyes. Right, we both have eyes, but the common ancestor that insects and vertebrates share did not have eyes. Right, so that similarity, in having eyes, is not due to the fact that they share a common ancestor. Instead, insects and vertebrates in a different time and a different place both separately evolved eyes. Wings are another striking example And so we just referred to the example of a bat. Um, well, birds and insects also have wings, but each of these cases um, occurred independently in different times and places. And so birds evolved wings, some mammals evolved wings like bats, and insects evolve wings, right? Convergence. They converge on a similar phenotype in a different time and place because there were similarities in similar selection pressures in each of these groups. And so the common ancestor of mammals and birds did not have wings. Birds and mammals and insects evolved wings independently. The other thing to mention is that not all traits that appear originated through the process of natural selection. They aren't adaptive, right? Not all traits originated because they influence the fitness or the reproductive capabilities of a species. And so if we think that something is an adaptation of a species to a particular environment that came about through natural selection, we have to show that rather than assume it. And so there are a number of different traits that we have, for example, that probably have nothing to do with our survival and reproduction. They just appeared through other means. And so these traits are sometimes referred to as spandrels, or neutral traits that came about as a byproduct of maybe selection for something else. And so the spandrel was sort of outlined in the late 1970s in some research by a couple guys, Gould and Lewontin. And so they argued that if you have an archway, like a building with an archway, there's a space between the archways or at the top that exist for structural reasons. They exist as a byproduct of the arch being present, and frequently, historically, those spaces, because they were just empty spaces, were decorated. And so those decorations and those spaces, those spandrels, appeared as a byproduct of the arch being present. They weren't created for the purpose of, for any other purpose. And so we see the same thing in biology and traits. So the chin, the fact that we have a projecting mental eminence on our jawbone, on our mandible, has nothing to do with survival and reproduction. It could be it's hypothesized to be the byproduct of reduced face projection, face size in human evolution. Of all the different extinct human species that we'll talk about, Homo sapiens, our species, is the one with a chin. It doesn't do anything, though. Earlobes is another one, or a belly button, which is the byproduct of having a large umbilical cord. Right? It doesn't do anything. It didn't arise because it improved the fitness or the survival of our population. We also have traits that are vestigial. They're left over from a time where it was adaptive in ancestral species. And one of the really cool examples of a vestigial trait, a leftover trait, is the grasping reflex in babies. If you've ever been around babies, or if you have family members who have them, or if you have had kids of your own, you might know that they're pretty strong, right? If you touch their hand or their foot, they will reflexively grab whatever the thing is that's there. And so this is hypothesized to be a leftover trait from when human ancestors were adapted to climbing trees millions of years ago. And we'll talk about what this group is called and how we know that they were adapted to tree climbing and those kinds of things in a few lectures. But so the idea is that if you are living in a tree and there's variation in the babies of this population, some of them reflexively grasp onto objects and others don't. The ones that reflexively grasped onto objects, right, you have the sensory sort of feedback and the tendency to do that in their hands and feet, were the ones that had a better chance of not falling from the tree. And so now we have this trait that doesn't really apply to us because we don't habitually live in trees as humans, but yet we still have it in our infants. Another example is the vestigial pelvis in whales, the pelvis bone. And so whales are aquatic animals, but whales and dolphins are actually mammals, right? So they have a streamlined body plan. They have fins instead of limbs, kind of like a shark. But dolphins and whales are more closely related to humans and other mammals like primates, like rodents, like carnivorans, like cats and dogs and bears and that kind of thing. Whales and dolphins are more closely related to those guys than they are to sharks. And that's because, so the vestigial pelvis in a whale is the remnant, it's the leftover pelvis from when the ancestor of the whale had lower limbs that attached to the pelvis. Another example of a vestigial trait in humans is the tailbone. We have a series of small fused vertebrae at the bottom of our vertebral column. And in fact, humans and all apes have that same trait. So humans, chimpanzees, gorillas, orangutans, and gibbons. We all have vestigial tailbones because the ancestor of apes had an external tail like other primates. And I'll talk about that in a little bit more detail as well. But the point is, not everything that we observe in an animal is an adaptation. Not everything that we observe appeared through the process of natural selection because it enhanced the survival and reproduction of that species. Sometimes traits are byproducts of other changes because of the effects of pleiotropy or integration, or because they haven't been eliminated yet, because there's no reason to, right? The vestigial pelvis in a whale hasn't been eliminated through the process of selection because it's not affecting the survival and reproduction of the whale. Okay, so again, I've mentioned that evolution works with what it has. It pushes things or pulls things in directions. And so closely related to the concept of an adaptation, where a trait arises through natural selection because it enhances the survival and reproduction of a species or individuals within that species or population, is the concept of exaptation. And that's where a trait evolves or appears for a reason or a function other than what it currently does. And so this is also pretty common among living things. One particularly interesting example of exaptation is the evolution and function of feathers. And so we see feathers obviously in birds, right? Things that can fly. And so we might wonder, is the evolution of feathers linked in some way to flight in birds? Well, it turns out that it's not. That an early form of feathers emerged in the ancestor of living birds in animals that did not have the ability to fly. And so you may not know that living birds, avians, are actually the living descendants of one of the dinosaur groups. And through the preservation, through the incredible preservation of dinosaur fossils in different parts of the world, we now know that some of them possessed early forms of feathers on their body. And so the evolution of feathers occurred in stages over time from a dinosaur ancestor that lived hundreds of millions of years ago. And only later in time, more recently in time, I'm thinking on the scale of tens of millions of years, did feathers play a role in the biology of flying birds, of avians. And so actually with the new Jurassic Park movies, Jurassic World or whatever, there was a push online for them to modify the dinosaurs to include feathers. Because when Jurassic Park was made in the 90s, this kind of thing wasn't as well understood. So like it's probably the case that like many of the dinosaurs that we think about as like scaled, reptile like things actually had bodies covered with feathers in some way. Okay, so that's exaptation, right? Feathers in a bird first appeared in ancestors for a function that's probably related to regulating their body temperature rather than having anything to do with flight. And that's because evolution works with what it has. It can take the variation that's preserved in this ancestor and shape it over time through the evolutionary process, right? Through natural selection to look more like this. Okay, so when we think about selection, there are different types or different modes of selection. And the first one is called directional selection, where a trait in a population is pushed in one particular direction, or an extreme trait is favored. And so if you have something like this bell-shaped thing, this curve, refers to a distribution of traits in a population. So this is a trait value of a continuous trait. We can think of this as like height or something, where these are the small individuals and these are the large individuals. This is the number of individuals, right? So most individuals, in other words, in this case, would have this trait value. And so if individuals on the extreme end of the spectrum had increased fitness, then those individuals would survive and reproduce and pass on those genes for that trait to their offspring. And so over time, there would be a shift. There would be a directional change from one trait average to a higher trait average. That's directional selection. There are lots of examples of directional selection. Here's a really cool example of directional selection that was observed over a short period of time in a wild population of finches, of birds. And so here are two different species of finch, both of which have variation in beak size in their population. And beak size is determines the size of the seeds that these individuals can eat. And so what happened was, in 1976, there was a major drought. And these birds live on islands, I should mention that. There was a major drought in 1976, which meant that only large seeds were available to eat. And so the individuals that had smaller beaks were less able to eat food, to consume food, which they need to survive. And so individuals with larger beaks had increased fitness. They had increased survival and reproduction. They had kids. They laid their eggs and had offspring that inherited those genes for larger beaks, which meant that in the following years, there was an increase in beak size observed in these populations, right? In other words, there was a directional change from this trait value in 1976. There was a directional change to these trait values in later years. That's an example of a small-scale microevolutionary pattern. And you can see clearly from years there's change over time in beak and other traits, beak size and other traits. So that's directional selection. We can also observe directional selection on a huge time scale, on a macroevolutionary time scale. And this is an example of brain size evolution over human evolutionary history. And so this is hominin brain size. Hominin being the term that refers to humans and our close relatives. We can observe, based on all of these fossils, each dot is a different fossil that goes back to about 7 million years ago. We'll talk about what each of these fossils is and how we know how old they are and many other aspects of their paleobiology. But we observe this directional increase between about 2.5 and 2 million years ago in brain size and body size in human evolutionary history. And again, each of these is a different fossil individual that represents different species over time. And so this is us today. This is males and females today. These are like Neanderthals and archaic humans in the past. This is Homo erectus. This is probably early Homo. This is Australopithecus and Paranthropus, and so on. We'll talk about that in a later lecture. So again, directional selection, where an extreme of a trait is favored. In this case, extreme brain size was favored. We'll talk about why that is. In this case, large beak size was favored on a short period of time or short time scale. The next form of selection is stabilizing selection, where the average trait is favored, where the individuals closest to this average value have a better chance at surviving and reproducing. And so that means that individuals on the high end and the low end do worse at surviving and reproducing. And so over time, it increases the number of individuals with that average trait, and it decreases the individuals at the extremes, right? These individuals don't survive and reproduce. And so only these individuals have surviving offspring that inherit their genes that code for this average value. So there's a fascinating example of this in humans, which is stabilizing selection on human birth weight, right? The weight of babies at the time of birth. And so we'll learn that humans are sort of unique among our close primate relatives in having both large body size of infants and, as the species we are, having a very large brain size compared to the size of our body. And so human babies have to fit a large brain and a large body through the birth canal. And so what we see is that individuals at about this birth weight have the easiest time of surviving. Now, it doesn't mean that you can't be smaller or larger than that at birth and not survive, but it means that as babies get increasingly larger, it becomes increasingly difficult for childbirth because they physically cannot fit through the bony birth canal. And so we're not going to see babies born that are 15 or 20 pounds. It's just physically not possible. Now, with the advent of modern medicine, C-sections are a thing where it's more of a surgical procedure, and of course that helps humans sort of get around these problems. But still, I mean, for the vast majority of human evolutionary history, this has been a thing that humans have had to deal with. On the other hand, if babies are born very, very small in birth weight, it means that they're susceptible to disease. And usually if they're born at such a small birth weight, it's because they're born premature. And that also affects their ability to survive. And so what we see is that the curve of the distribution of human birth weights is exactly flipped. It's exactly opposite the mortality rate. And so, in other words, individuals at this end of the mortality rate profile have the lowest chance of dying and the highest chance of surviving, whereas these individuals at low and high birth weights have higher chances of not surviving. And so the extremes, the low birth weights and the high birth weights, are not favored by natural selection. Instead, individuals in the middle, around this weight, have the highest evolutionary fitness. And so selection allows babies to be born at that size. Okay, the third type of selection, or mode of selection, is called disruptive selection. And this is where both extremes are favored. And so if you start out with a population that has some trait that has this distribution in the population, over time, because individuals at the high end and the low end are favored, individuals in the middle have lower fitness, and this creates what's called a bimodal distribution, where you've got two peaks. Some individuals have this trait, and other individuals have this trait. And so here's a hypothetical example. This plot is showing Arctic temperature over time. And so basically what we see is that things are heating up, things are warming up, which leads to decreasing snowfall in this hypothetical example and a rockier environment where the white and gray and black rabbit blend in better. So let's say you go from something like this to something like this. Now you have these patches of lightly colored areas with patches of dark colored areas. So in this case, you might expect an original population of white rabbits to, through the process of disruptive selection, to produce two new populations. One where the darker colored rabbits blend in with the darker spots more easily and therefore survive and are protected from predators and other issues. And another population where light colored rabbits are favored in areas in this environment that are lightly colored and are therefore camouflaged. So that's an example of disruptive selection. Okay, now we've talked about natural selection. We've talked about some of the experiments that have been done, like the fox experiment, which is a form of artificial selection where humans are doing the selecting. But there's also something, a third form of selection called sexual selection. And this is where you've got natural selection acting on traits that influence mating success. And this is really interesting because under sexual selection, you have the evolution of traits that influence mating success within the biological sexes of a species, right? So among females and among males. Okay, so in most mammals, there's a mismatch in reproductive investment. So this is a sperm and egg. This is like a kind of like an analogy for the reproductive investment made by males and females. In most species, females invest more resources into reproduction than males do. And that's because in females, because of the way that sexual reproduction works, females in different species are the ones that gestate, right? The offspring develops and grows within those individuals. And as mammals are defined by having mammary glands and the ability to feed offspring through milk. And so the females among mammals are also sort of tasked with feeding offspring because the males cannot produce milk. And so females invest more resources than males, right? Gestation and lactation takes time. And so the time required, the time investment in offspring for females means that they can have fewer offspring. And so each one sort of counts more for females among mammals than it does for males. And that's because male reproductive output is not limited in the same way that female reproductive output is. And so the sex with more limited reproductive potential should be competed over by the sex with the greater reproductive potential. And so this is a graph showing the number of offspring produced against the number of mates. And so the idea is that in mammals, as the number of mates increases, so does the number of offspring. Right? So if male individuals want to have more offspring, they just have to have more mates. In females, there's a different pattern where once you have one successful mating, additional mates does not increase the number of offspring produced because of gestation and lactation. And so we can define a couple of terms here. The first is reproductive potential. That's the total possible output of offspring by one sex. And the other is reproductive variance, which is the variation in reproductive success within a sex. So variation or variance you can think of as like, do all males have the same number of kids? Do all females have the same number of kids? Or are there some males with 10, 20, 30 kids and some males with 0 or 1? Are there some females with 0 or 1 kids, or are there some females with 30, 40, 50 kids? So that's what we mean by variance. The limiting sex then has low reproductive variance and low reproductive potential. Low reproductive variance meaning that whatever the limiting sex is, there's not that much difference from person to person or individual to individual in the number of offspring. The non-limiting sex has high reproductive variance and high reproductive potential. So that means that some individuals have a lot of kids, some individuals have no kids, and some individuals are in the middle. And so Bateman's principle states that in most species, reproductive variance is greater in males than in females. It's not the case that some females have 50 offspring and other females have 0. If females have offspring, usually the number of offspring is somewhere around the same number. Whereas in males, it's not uncommon to have some males that have a lot of offspring and other males that have very few. And so here's some example data of the maximum number of offspring produced during the lifetime of males and females in different species. And so you've got an elephant seal. The max number recorded is 100. The max for female is 8. In the red deer, the max is 24 for male and 14 for female. In this bird, the max is 26 for the male and 28 for the female. And this is important because birds are not mammals, right? So birds don't have the same relationship between male and female reproductive output that mammals do. So what do you think the max number for a human male is during lifetime? What do you think that number is for a female? Like, take a second to think about what those numbers might be. $888 for a male and 69 for a female. So number one, clearly those are different numbers. But number two, the actual number for males and females to me is like astounding. I mean, even having 69 offspring for female is like almost incomprehensible. But the point is, the number of offspring that males can, that males can have among mammals is higher than the number of offspring that females can have in mammals within species, that's because females have a greater reproductive investment than males do. And so there's sort of different ways that we can think about sexual selection. And the first one is female preference and male display, where females are choosing certain males that have traits that are desirable in some way. And that produces something called sexual dimorphism, or sexual selection in general produces something called sexual dimorphism. It means like multiple morphs, multiple varieties, right? Males and females look different. And so this is a male bird, and that's a female bird. And so in birds, this is not an uncommon pattern where the males will have sort of like different decorations or different colorations of their feathers. And that's because they use those things and those traits to display to females, who then choose a mate. And so they choose the mate with the brilliantly colored feathers, and their offspring therefore inherits those brilliantly colored feathers, which helps their offspring have more kids, and so on and so forth. And so that's an example of what's called intersexual selection, the evolution of traits that increase the chance of being chosen as a mate, for example, through the process of female choice. And so in birds, males evolve these displays and these crazy colorations of their feathers so that it increases the chance of a female choosing them to mate. And so this is an example of a male in some bird species, I don't know the species actually, that has this particular mating display where the feathers get pushed out, and they have this kind of funny pattern of black and blue on their feathers, whereas the females don't have the same kind of phenotype. And that's because females are choosing males with these particular traits. We also have male-male competition. And so the thing that, as we've mentioned about, the thing that limits the number of offspring that male mammals can produce is access to females. And so males will compete directly and indirectly with each other within a species for access to female individuals. And so in response to male-male competition, usually we see the evolution in mammals of body size dimorphism, body size increase, where males evolve larger body mass. We see the evolution of weaponry. In primates, that's in the form of canine teeth, right? So here are the large canines of a baboon, the large body size of the baboon in the male individuals compared to adult females. You see the evolution of horns and antlers and that kind of thing in different mammal species. Those are other examples of weaponry because it helps them win competitions. And we refer to this as intra-sexual selection, where traits evolve in response to competition between members within a sex. So that's male-male competition. And so basically the way it works is that if you have competition between males, like fighting between males, usually, and this is in a primate sort of example, if males are winning those fights, they assume a higher rank in a dominance hierarchy. And so usually the higher the rank in the dominance hierarchy, the more the greater the number of female mates. And so this is what this data is showing here. Basically, the higher the rank in the dominance hierarchy, the greater the number of female mates. As rank decreases, as you lose more and more fights, eventually things just even out, and these individuals have very few mates. And so if an individual's ability to win those contests is based on some trait that they produce, like the large canine teeth of the baboon or the horns or antlers of other mammals. If that helps them um, increase their number of mates, then that increases the number of potential offspring they have, therefore increasing their evolutionary fitness. And so there's a greater chance that they'll pass on those large canines or their large body size or their horns and antlers to the offspring that they produce with those increased number of mates. We also have the opposite example where females compete for males. This is a more uncommon example, but there's a cool group of New World monkeys, New World primates in South America, where males take care of young individuals. The females compete over males, and interestingly enough, these species have a high frequency of twinning, of giving birth to twins. And so this is kind of like the unique primate exception to the rule. Okay. One thing I want to mention before we move on is that, like, when we're thinking about like male competition and female choice and that kind of thing, again, like we usually think about humans, right? Like you're probably thinking, oh, how does this play out in humans? Humans are not immune from the evolutionary process, and we may have things in our culture that appear to approximate some of these things that we're talking about, but it's not as clear cut in humans. And that's because humans have culture. Humans have culture, and humans have ideas about gender, which differ from biological sex. And so humans take all of these things to the sort of next level. And in humans, we see kind of like every example on the spectrum that we can think of. And so humans, again, we're not immune to the evolutionary process. We're still animals. We still have a biology. But at the same time, our culture, which varies in different parts of the world, acts as a buffer against some of these things. So just be mindful of that. Don't think that just because we see this in a baboon or a beetle or an elephant seal that it means that it applies directly to humans, at least without further inspection. Okay. So here are the 4 forces of evolution, right? Remember what the 4 forces are and why they matter. So here's a kind of summary. Mutation, gene flow, genetic drift, and selection. Mutation, gene flow, and genetic drift are random or quasi-random processes. In other words, there's no reason that one allele is favored over another or one trait is favored over another in these evolutionary processes. However, in selection, whether that be artificial, natural selection, or sexual selection, the process is not random. And that's because certain alleles influence fitness, certain traits influence fitness, and that is not random. Mutation increases variation within populations. That's because new alleles can emerge through mutation. You can add new genetic variants. You can add new genes to the population. Gene flow increases variation in your population because new genes are flowing from one population to the focal population. Genetic drift decreases variation within your population because you're losing genetic variants, you're losing alleles over time. If alleles are evolving to fixation, it also is a decrease. It also implies a decrease in genetic variation because other alleles are not present, right? If one allele is at 100% in the population, it means that there aren't other individuals with different alleles. So therefore, it's a decrease in variation. Selection depends whether it increases or decreases variation within the population. If it's, let's say, stabilizing selection, it's going to decrease variation within the population. But at the same time, if you have disruptive selection, for example, it can increase variation in the population because it's favoring individuals at the extreme. Okay, now what about the differences between populations? Mutation increases the difference between populations. In other words, populations are evolving to be different through the process of mutation, right? They're becoming more and more different as mutations appear. Under gene flow, the difference between populations is decreasing. It's getting smaller. The two populations are becoming more similar because one population is sharing genetic variants or genes with another, right? So there's a decrease in population variation. Under genetic drift, populations become more different from each other because they are randomly losing alleles or randomly losing variation over time. And of course, selection is environment dependent. Populations can evolve to be more distinct from one another through selection, or they can evolve to be more similar to one another through selection. It just depends on the circumstances. Okay, so here is the way that I think about the four forces of evolution. Right, here's mutation, gene flow, drift, and selection. Under selection, you have three different forms: natural selection, artificial selection, and sexual selection. Natural selection is like your garden variety selection that we observe in nature. That's like the finch beaks evolving to be larger in response to the drought. That's human brain size increasing over time. Artificial selection is when humans do the selecting. That's like the fox experiment, or the domestication of dogs, or breeding plants for certain characteristics. And then you've got sexual selection, which is the evolution of traits that influence mating success. Under sexual selection, you have two forms: intrasexual selection within a sex. So like that's like the evolution of traits that improve one's ability to compete with individuals within your sex. Intrasexual selection. Intra means like within. So this is going to be usually male-male competition, the result of male-male competition. Intersexual selection is the evolution of traits that influence your probability, your chance of being chosen as a mate. And usually in mammals, or usually just in general, this is female choice. This is usually females choosing males with certain traits that are desirable for whatever reason. Okay, now we have different modes or models of selection where some quantitative trait can be pushed or pulled in a particular direction. That's directional selection. Where a trait can be stabilized around some average value, where the high and low value individuals are that they have a lower chance of surviving and reproducing, lower fitness. And then finally, disruptive selection, where extremes are favored and the average individuals are not favored. Okay, so I always end the evolutionary theory lecture with this list of things of what not to expect of evolution. Because people have a lot of misconceptions about what evolution is and what we should expect from it. So there's a lot of text here. I don't really mind it because you at least have all the text. This is from a textbook, a really good textbook called Evolution, if you ever want to look into this in more detail. So number one, we don't expect the necessity of adaptation We don't always think that animals or that living things should be evolving through the force of natural selection. Traits can be advantageous without being necessary for survival. So, not everything is an adaptation. Not everything that we can observe in an animal came about through selection. It could come about through mutation, through drift, through gene flow, right? It could emerge, it could appear through the other forces of evolution, other than natural selection. We don't expect perfection. The idea of perfection is the idea that somehow evolution is like pushing species in a particular direction. That evolution has some end goal. That it's like trying to make species, quote, better. So in this case, best traits, like the optimal best ever trait, can evolve if they don't already appear naturally in the population through mutation. Humans are not going to evolve wings because we don't have wing-like structures as part of our arms naturally. Right? A perfect evolutionary solution may never happen due to constraints. And so, again, we're not going to spontaneously evolve some ability to fly just because it would be cool, just because it would be better for us, right? It's not going to happen, right? So we don't expect perfection. Evolution is just change. It's not change in any particular direction necessarily. At the same time, under that same kind of way of thinking, we observe frequently traits that appeared through the evolutionary process, but have negative consequences, right? So humans walk on two legs, or bipedal instead of quadrupedal, like other animals, most other animals, and so because of that, guess what? Now we have ankle problems, knee problems, hip problems, and back problems as we age. And that's a scar, a so-called scar of evolution. When you are 80 years old and your knee hurts because you're bipedal and you've been walking on your knee joints for 80 years, it has no effect. That deleterious side effect has no effect on your evolutionary fitness. Because if you're 80 years old, chances are you've probably had all the offspring that you'll ever hope to have at that point. And so, not everything that an organism possesses is like some perfect thing, right? We're just biological beings trying to survive and reproduce. And through that survival and reproduction, things change. Gene frequencies change. The phenotype changes. It doesn't mean we're becoming more perfect. It just means that we're changing. And so on that, the last thing I'll say is humans, just because we are cognitively advanced, just because we have a high level of cognition and written language and modern culture and technology, doesn't mean that we are more perfected than any other species. It doesn't mean that we are, quote, more evolved than chimpanzees, our closest living relative, right? So if you've thought about evolution in that way, eliminate that from your thinking. That's wrong. Humans are not more evolved. Chimpanzees are not less evolved, right? Humans and chimpanzees have been evolving for the same amount of time, 6 or 7 million years. We've just evolved differently. Now, you may interpret the differences in human and chimpanzee evolution as being better for us, and maybe in some ways they are, but that's a subjective interpretation, right? Species are not trying to evolve large brain size. There's no trying, right? There's no other force that pushes species in one direction or another. It's only their survival and reproduction. The only try is trying to survive. We don't expect progress. because progress implies that there's some goal. This is sort of what I was just talking about. There's no end goal, there's no ideal, right? Fish are not trying to evolve limbs. Nobody's trying to evolve anything. After the fact, after the evolutionary change happens, we can look back and say, oh, a trend happened where some trait increased over time, like we talked about with brain size. Oh, brain size increased over the last 2.5 million years in human evolutionary history. That's not because 2 million years ago they were like, man, how can we get access to the nutrition to help our brain size evolve? No, they were just trying to survive, and the individuals with larger brain sizes were the ones that had more kids than the individuals with small brain sizes. That's it. There's no thing pushing hominins to have large brain size, right? There's no external force. It's just them surviving. We don't expect harmony and the balance of nature, right? So organisms, genes are inherently selfish, right? So if we see like this nice, harmonious thing in nature, it may look nice. We may interpret it that way. But the truth is, cooperative behaviors are beneficial to individuals. If it was not beneficial to individuals, it would not evolve. And so, yes, doing things for other individuals, I'm not talking about humans necessarily, but cooperation among animals is the result of the evolutionary process, the selfish evolution of those organisms and genes. If you have some balance in nature, it's the result of the selfish evolution of its constituents, the plants and animals. That's it. Now, it doesn't mean that as humans, we can't look at those things and enjoy them or appreciate them. It doesn't mean that we can't choose to act cooperatively and altruistically, but it means that we don't explain it through like some harmonious kind of thing. It's just the evolutionary process, right? It's just change over time. That's it. And finally, and this may be the most important one of all, we don't expect morality and ethics to emerge from the evolutionary process. There's nothing moral or immoral to anything produced by evolution, right? What's right and what's wrong has nothing to do with nature. It has nothing to do with what animals do, right? What's right and what's wrong is determined by us in our shared sort of cultural world. And so there's a long, nasty history of people misusing this idea that just because you see something in nature means that it's desirable or good, right? Or that we should use the evolutionary process as a way to like modify humans. And so like eugenics, for example, is a result of this flawed thinking, right? That somehow, humans are just part of the natural world. Animals do this, therefore we should be doing this too. No, that is not the case. And so this is referred to as the naturalistic fallacy. The idea that just because something is found in nature, just because something is natural, means that it's good, right? Well, there's plenty of stuff that we find in nature that is totally awful and undesirable, right? There's no legitimate philosophical foundation for the perspective that just because fish do this means that we should adopt it. And so this is a list of what I would think is a series of popular kind of misconceptions about what evolution is. Evolution is just change in populations over time. That's it. There's nothing perfect about it. It's not like species are trying to get to some other end point. It's not that, humans are more evolved than fish. It's just biological change over time. Okay. Let's talk about the lab report that's due on Thursday. I uploaded a data set and talked a little bit about the lab up until this point. In the lab, you will have to analyze data to test hypotheses. So let's talk about different types of data first. There are two major types of data. Some data are qualitative, and some data are quantitative. I think when you think of data, if you've ever thought of data, you're probably usually thinking of quantitative data, like measurements of stuff. But that's not the only kind of data. A variable is refers to the different properties that are measured in some way. So qualitative data is also known as categorical data, and these are descriptive, non-numerical data. So categories, right? Like, is an individual biological male or female in terms of their biological sex? What season is it? Summer, fall, winter, spring, etc. Coloration, red, green, blue, whatever. Those are qualitative data. Wait, we've not measured any like continuous thing about this. Quantitative data, on the other hand, are numerical. There's two forms. There's numerical and continuous data. So numerical data consists of discrete or discontinuous whole numbers, or like count data, like meristic data. So like, for example, in the human spine, in the vertebral column, we have different kinds of vertebrae, right? Certain neck vertebrae, those are cervical. Thoracic vertebrae, those are part of the thorax and the rib cage. And lumbar vertebrae, those are in the lower back. And then below lumbar vertebrae, we have sacral vertebrae as part of the sacrum, and coccygeal vertebrae, which are just a few fused vertebrae in humans. So you could count the different numbers of vertebrae in each human, and there's going to be some variation. Some humans have 5 lumbar vertebrae, some humans have 6 lumbar vertebrae. That would be an example of meristic data or count data. At the same time, we also have continuous data. And continuous data refers to numbers that can take on an infinite range, even within a fixed range. So height might be one example. You could be 0 feet tall, not really, but that's hypothetically possible, or 0.1 feet tall or whatever, all the way up into infinity. So continuous data are what we are dealing with in this lab. We also have to define, when we think about analysis and statistics, the difference between a population and a sample. A statistical population is the set of all possible individuals, objects, or units that might have been included in your data. So for example, you could say, what is the mean height of humans? Well, hypothetically, we could measure the height of every single individual of the species Homo sapiens that's ever lived, every adult individual, let's say at the age of 20, and we could tell you exactly what the mean value is of the height for every single Homo sapiens individual through all of time that's ever lived. Well, we really can't do that. It's really not possible. And that's in part because we don't have the skeleton of every individual that's ever lived. We can't go back in time, right? So it's not realistic to characterize the population. So instead, what we do is we collect a sample, which is a selection of cases from a population. So we can't measure every single human being, but we can measure hundreds or thousands of people. And so when we measure a sample, we intend for the sample to approximate the population in some sense, right? In other words, that like the mean or the average height of our human sample is reasonably close to the mean of the population. But sampling is random. Ideally, frequently though, when we sample populations, there's a bias. The sample is not picked at random. And so, for example, when we analyze heights of students from the data set that I uploaded to Blackboard, that's not really a random sample of the human population, is it? No, it's a sample of students from Hunter College, which live in a certain part of the world, which are a certain age, right? It's not a random sample of all different kinds of people everywhere around the world. Now, that it turns out that bias may not be a huge deal. The average height for the people in the data set is probably a reasonable approximation of the population of all individuals within that age range around the world. It's not going to be hugely off. How much that difference is and whether it matters is another question. Okay, so if we could measure every individual in the population, we could characterize what's called a parameter. That's a statistic about the population. So again, like using our height example, we could quantify the population parameter that is equivalent to the mean, right? The population mean. And that would tell us with no error, right, with certainty, what the mean value is for all human heights through all of time and space. But again, we don't know that population parameter. And so we have to infer that value from our sample. And so what we use is a sampling statistic, or just a statistic. And that is a statistic about a sample. And so when you see the word statistics or statistic, this is what we're talking about. We're talking about some characterization of a sample of a population. So let's talk about a few statistics that you probably already know about. These are descriptive statistics because they describe a sample. And three of them are each kind of averages. These are all kind of an average. The first one is the mean. That's what we usually think of as the average. It's the sum of values, right? You add up all the values in a data set and you divide by the number of values. So the average or the mean height of the data set we have is all of the heights added up divided by the number of students. The median is the middle value. It's the value that divides the distribution of data or the distribution of values into equal portions. The mode is the most frequent value. So if the distribution of your data, of your height data, or the income data in this case, have the same mean and median, it means it's a normal distribution, a quote normal distribution. And we don't mean normal as in, oh, it's just a normal distribution. No, it's referring to a very specific thing. It's a normal distribution, right? It's a bell-shaped distribution that looks something like that, where the mean and the median are the same number. However, Not all distributions are normal. And if it's not normal, then the mean and the median will be very different. If they are very different, then the distribution is skewed. It's no longer normal. And so income is a good example of a distribution that is not normally distributed. And that's because under a normal distribution, let's say that this is a normal distribution. It means that there are the same number of individuals that have less income than the mean compared to the same number of individuals that has more income than the mean. And with income, that's not the case. In terms of income, there are more individuals that have less than the mean than there are individuals that have more than the mean. So in other words, there are a few people who have an astronomical amount of money on this end of the spectrum. There are very few people that have a lot of money in that way compared to many more individuals who have less income. So in this case, if you take the average, the mean of this sample, the number of individuals at this end are going to pull the mean in this direction. So the mean is going to be right here. Right? The mean is influenced by these kind of high outlier individuals, whereas the median will be lower. And so usually when you're looking at income or you're looking at data sets that have outliers, the median is usually a useful thing to look at. So for example, if you're looking at like the average, if you're looking at like the cost of buying a house, or if you're looking at how much rent is in a particular neighborhood, it usually doesn't help to look at the mean. It usually helps to look at the median. And that's because there's going to be a small number of individuals who have really, really expensive houses or have really, really high rent that pulls the number up. So when you're looking at anything like that, just in your own life, look at both the mean and the median. Okay, so if we have some data, we can look at the mean, the median, and the mode of height, for example. But we also have a number of what are called measures of spread. And the first one is called the standard deviation. And that's just the average deviation from the mean. How much does each individual in the data set differ from the mean? If there's a huge variation, like some individuals are super, super, super tall, other individuals are super, super, super short, then they're going to differ a lot from the mean value. And so the standard deviation then is going to be increased. The variance is just standard deviation squared, right? So that just means standard deviation, whatever that number is, multiplied by standard deviation. And so if you have, let's say, a normal distribution with high standard deviation, it's going to look something like this, where you got some mean value and the spread around that is quite high. If instead, you could also have a normal distribution with the same mean value here, but with a much reduced spread of the data around that mean. And so in this case, the standard deviation of this sample will be lower than the standard deviation of this sample. The variance of this sample will be lower than the variance of this sample. And so for normal distributions, we have this pattern that emerges where 68% of individuals fall within one standard deviation of the mean. 95% of individuals fall within two standard deviations of the mean. 99% of individuals will fall within three standard deviations from the mean. Anything higher than that, this would be 1% of individuals. So 0.5% and 0.5% of individuals will be 4 standard deviations from the mean. So those are measures of spread. We can visualize quantitative data in a number of different ways. If you have one variable, this is an age variable, you can use something called a histogram. And a histogram is a graphical display of what are called binned frequencies of continuous data. All right, what the hell does that mean? Okay, it basically just means that you take the range of whatever you're looking at, so age 0 to 100, and you divide them into categories. You say like, all right, how many individuals are 30? How many individuals are 25? How many individuals are, what did I do? Yeah, 30, 25, 20, 15, right? And you just count the number of individuals in each of those groups. The bins can be, instead of just age, like age 30, age 25, age 20, it could be like how many individuals are between age 20 to 25, or 25 to 30, or 30 to 40, right? You can decide what the bins are. And so on the x-axis is the bins, the categories. In this case, each category is just individuals within that age range. And on the y-axis is the number of individuals. So this is the peak. So most individuals are within this bin. And so here, the mean is 36.6 years of age for this sample, with a standard deviation of 22.5 years. So that means that the average difference of each individual, each of these 132,000 individuals, the average difference between each individual age and the average of 36.6 is 22.5. Each individual is, on average, 22.5 years below or above that average. Okay. So we can represent large data sets with histograms. And so it goes like this. You first divide your data into bins, into intervals, and you just count the frequency. And so we can look at, let's say, midterm test scores. You know, one person got a 70, one got an 81, a 74, a 99, an 83, and so on. And we can decide that our bins or our intervals will be like 5% bins, 5-score bins. So 65 to 70, 70 to 75, 75 to 80, right, all the way to 100. And then we just count the number of individuals within each bin. So of this set of test scores, how many individuals fall between 65 and 70 in terms of their score? Well, it's just one. And we count, in this case, we count 70 in this bin. How many individuals fall between 70 and 75? If we go through, hey, just one. So we put a 1 here. How many individuals fall between 75 and 80? So there's 74, 81, 0. What about 80 to 85? Right, we can do that for every single individual, and then we can plot this categorization of our data as a histogram, right? So again, on the x-axis here, you have bins, 65 to 70, 70 to 75, and then you have the number of individuals, which you can call frequency. So we have one individual in this bin, one individual in this bin, 0 individuals in this bin, 2 individuals in this bin, and so on. And so if you have a huge data set, it looks like this. That's a histogram. But what if you have more than one variable? Right? We looked at test scores, we looked at age. What if you have 2 variables and your study or your analysis has a specific hypothesis about how those two things are connected, well, we can visualize those variables with a scatter plot. So a scatter plot is just a display of sets of more than one variable. In this case, we think they are linked. So here's some data on the number of beers drank the night before an exam. And on the y-axis is the grade on the exam. And so we note that there appears to be a trend, a relationship, that as the number of beers drank increases from 0 to 15, the exam scores decrease from 95 to about 70. And so in other words, we think these two things are correlated. And correlation just refers to the departure of two random variables from independence, or the extent to which one variable predicts another. And so in this case, we might suggest that the number of beers drank the night before an exam predicts what the grade will be on that exam. And so there are a number of ways to measure the correlation or measure the predictive power of one variable for another. And so if you have a scatter plot of data, we can fit a regression line or a linear model to those data. And so this is a theoretical line of best fit that minimizes the distance between each point and the line. And this characterizes the relationship between two variables. And so essentially, there are different ways to fit this line, different approaches to regression. We're not going to go into that. But you might have recognized from previous math class this old trusty thing, this formula y equals mx plus b. And so the y refers to the y variable. The m refers to the slope of this line. The x refers to this variable. And b refers to the y-intercept, or the spot where the line crosses the y-axis. So if you can characterize the relationship of these two things, or these two variables, if you have a new case, let's say, hey, next year, individuals will still engage in the same activity before an exam, or let's say the next exam, based on the number of beers drank the night before, we can predict what the exam score will be. We can plug in that number to our equation that characterizes this relationship to make a prediction about what the grade will be. So regression is like at the core of statistics. When you use Google Maps to get directions from one place to another, it tells you what your estimated time is. Well, what do you think the estimated time is? How are they estimating that? Right? They're making a prediction based on the model. Just like we can make a prediction about what your exam grade will be, or what a student's exam grade will be the day after they drank a bunch of beer. Okay. So as I said, we can characterize the predictive power and the relationship between variables with a couple of different statistics. And the first one is called the coefficient of determination. Coefficient of determination, right? We can just call it R squared. It's the extent to which the regression line characterizes the relationship between two variables. And so usually there's like an arbitrary point where you say anything that's like 0.7 or higher is a strong correlation. Yes, that's true, but if the r squared is lower than that, it doesn't mean that it's not correlated. It just means that it's explaining less variation. So in this case, let's say the r squared value, this is just means caret, it just means it should be up here. The r squared value is 0.55. It means that the number of beers drank the night before the exam accounts for or explains 55% of the variation in grade. So it doesn't explain 100% of the variation in grade. If all the points fell exactly on the line, it would explain more variation in grade. So you can imagine that there are other factors that also contribute to the grade a student gets. It's number of hours studied, number of lectures missed, experience in a particular topic, and a variety of other factors, right? Memory, right? And we can think of a number of things that explain the other parts of what grade the student gets. The other statistic is just an r with no r squared, and this is the correlation coefficient. And so r is the value you use to draw real-life conclusions from a regression. And so in this case, because the relationship between these two variables is negative, it means that as one variable goes up, the x variable increases, the y variable decreases, right? As number of beers drank increases, exam grade decreases. A positive relationship would look like this. You'd have a scatter of points that look like this. And so in this case, because it's a negative relationship, the r value, the correlation coefficient, is a negative value. So in this case, it's negative 0.74. And it means that if someone ranks one standard deviation above average in beers drank, they will rank 0.74 standard deviations below average in the exam grade. So it just means that if you drink more, if one student drinks more beers than the average student everywhere the night before the exam, they will be 0.74 standard deviations below average in beers drank. Okay, so here are all the things that you have to do for the lab report. You got to follow the formatting instructions. You have to include all the sections: introduction, predictions, /hypotheses, methods, results, discussion, etc. You have to include figures that are labeled correctly. And then your writing has to be reasonably clear, right? It has to make sense. So the lab report is 2 pages long, total, including figures, which means you have to be concise. You can't just ramble on and on. Anything longer than 2 pages is not good. It means you get less points. The font is 12-point Times New Roman. It's not 13-point Arial. It's not 10-point Helvetica. It's 12-point Times New Roman. Single spaced, not 1.1 spaced, not 1.5, not 2. And 1-inch margins. Again, not 1.1 inch, not 1.5, 1 inch. These are the different sections. Introduction, which includes hypotheses and predictions. Methods, which includes data collection, how the data were collected. And data analysis, what you did to analyze the data. The results, what the analysis shows. And then the discussion, what it means. And these are all of the sections that you would find in a typical scientific article, right? An article that a scientist produces that goes through the peer review process and is published in an academic journal. These publications are how results are shared with other researchers and with the public. So let's say somebody creates a new drug to treat a disease. Let's say a successful coronavirus vaccine is created and tested. It will be presented to the public, to the government, to researchers through a scientific article that other scientists and experts review just to check that the results are accurate. If you're a political scientist or an anthropologist or a psychologist or a computer scientist or a nutritionist, those different fields will also produce research and produce these articles. These are the articles that I write on human evolution or on new fossils, right? So this is a part of the process of doing science. This is everything for research. Okay, so the intro introduces the topic. You're trying to explain why this research makes sense, why it makes sense to ask this particular question. You're stating your specific focus for the research, and then briefly mention how you examined it. And so usually we use this analogy, like this funnel analogy, where you start out really general, like genetics are important, phenotype is important. Supporting statements, you get a little bit more specific. Genetics play a role in multiple factors of human life and animal life. Phenotype has been, let's say, a focus of biological and evolutionary studies for decades. Then you have some specific statement. In this study, we evaluate the effects of genetics and environment on phenotype by examining height, grip strength, and limb proportions, let's say. Hypotheses and predictions should clearly state each hypothesis, the 3 hypotheses that you developed for the Lab 1.1 worksheet. You can give the direction of the expected relationship if that makes sense. I expect a positive relationship between these 2 things or a negative relationship between these 2 things. Remember, this doesn't mean whether it exists. We're not saying like positive relationship means that there is a relationship. It means that does the number of beers drank improve or decrease your exam grade? Should there be a strong connection or correlation between the 2 variables? And so on. You can also give the justification for the expected relationship if that makes sense. It just depends on the particular hypothesis. The methods describe the data collected. You can describe the type of data, like what were the variables. You can state the sample size. Of course, the data you can mention, and this is important. You should mention that the data were collected by previous classes over the last few years, and we're not collecting data because we're doing this remotely. And so the methods, the purpose of this section is so, number one, scientists and researchers and the members of the public can understand how you came about your conclusions, how you got the data to support your conclusions, and so that somebody can repeat the study. So somebody should be able to read your methods section, and you've written it clearly enough that they can then separately repeat what you did and get the same result or not. If they don't get the same result, then they can publish that article and challenge your conclusion, right? That's how science works. The analysis should describe how you calculated any new variables from the collected data, right? You don't just say, I use the Excel template. Like if you're using average parental height, you have to say this is an average that was calculated from the height of the mother and the father by adding those 2 values together and dividing that by 2, which is the number of individuals. Don't just say, I calculated in Excel. Talk about the statistical methods you use to analyze the data in simple terms, right? We're not statisticians. We're just saying we used Microsoft Excel or Google Sheets to analyze the relationship between 2 variables, etc. The results state what you found for each hypothesis. You get this from your analysis and your figures. and you should reference the figure. You should say, okay, for the first hypothesis, I analyzed this and I found that. Figure 1 shows the relationship between these, or does not show the relationship between these, or whatever it might be. And then you should mention that whether you support or refute your hypothesis. Figures are pictures, right? They're images in a scientific article that shows the data, right? It shows the analysis that you performed. It should be large enough, with large enough font and symbols that we can read it. Like if these are so small that you can't even see what the data show, then it's not useful. The variables have to be correct on each axis, on the y-axis and x-axis, and labeled properly, right? So you should have a title with units. So average parental height, for example, in centimeters. The units are really important because without the units, you can't interpret the correlation coefficient. You can't interpret the slope of this line, etc. You don't need to put a title here. The title is kind of redundant. You should also include in your figures where you use regression like this. You should include the r-squared value and the trend line. So here's the r-squared value. Each figure has a caption, has a legend. Each figure is labeled figure 1, figure 2, and figure 3. And so each figure should be a test of each of your hypotheses. You should include this concise description of what is presented in your figure, including units. So the figure 1 would say, it would just say correlation between student and average parent height in centimeters. Student height is weakly and positively correlated with average parent height in this case. So again, you're kind of like summarizing as simply and as briefly as possible what it shows. The discussion discusses the results that you obtained in the context of like what the field is, in the field of genetics and phenotype and all of that kind of thing. What does it mean for the big picture? Now, obviously, as students, we have a limited perspective on this, but we know enough now through the readings in the lecture to talk a little bit about how this matters for this topic. You can also talk about possible problems or limitations or sources of error in your data and analysis. So is there anything about the data or the data collection that influences the results? There might be. So maybe talk about that. And then you can summarize how your results fit into what else we know on the subject. You can refer to lectures and readings sort of as like the literature part of the discussion. It's important to know the Hunter College policy on academic integrity. This is here for you to read. You should know this by now from all courses, right? So don't plagiarize. Don't just take text from other things. You have to use your own words to do this, right? That's academic dishonesty if not, it's cheating if not, and it has significant consequences. Okay, so here's an example of a hypothesis for the first question that we talked about in that 1.1 worksheet. That H1, usually hypotheses are written like this, hypothesis _1. Offspring height is influenced by the genetics inherited from biological parents. And so this hypothesis might have a prediction that student height should be positively correlated with average parental height. And so we can use Microsoft Excel or Google Sheets to create a scatter plot that we just talked about to visualize the relationship between student height on the y-axis and average parental height on the x-axis. And so you'll have to include a scatter plot, a trend line, and the r-squared value for this analysis in your figure. And I'll show you how to do that. H2, human populations have been locally adapted to climate by evolving longer legs in warmer climates and shorter legs in colder climates. This is also known as Allen's rule. So the prediction might be that latitude is significantly and negatively correlated with relative leg length, which means that there are longer legs in lower latitudes and shorter legs in higher latitudes. So we'll use Excel or Google Sheets to create a scatter plot to visualize the relationship between relative leg length, which in our data sheet is labeled as seated divided by stand. So seated height divided by standing height on the y-axis, and latitude on the x-axis. H3 is that relatively long forearms increase grip strength. And so our prediction might be that relative forearm length is significantly and positively correlated with grip strength. So we'll use Excel or Google Sheets to create a third scatter plot to visualize the relationship between these two variables. Okay. So here is the Lab 1 report template. It has some additional instructions. There's instructions in the PowerPoint slide and the lecture here. And this is the template, right? So you'll put your name here. That's my name. The date of today. And you'll talk about the thing you're working on. Let's say the study of the relative roles of genetics and environment on human phenotype has long been a focus of biological studies. There's some redundancy because I said studies twice, but you get the point. Or you could just say, there you go, right? Don't use my sentence, obviously. Write your own sentence, right? So you've got the intro. Come on. All right, whatever. You have the intro, hypotheses and predictions. You can use the ones that I just provided, or you can use your own. The methods section is here, right? This is concise. You don't have a ton of space. The figures will be placed here, right? So you can copy and paste your figures from Sheets, Google Sheets, or Microsoft Excel. You can write a little caption here, and then you can write your results and your discussion here. What did you find? What do you think it means? Are there any problems with what you found? And so on. And then you'll upload this on Blackboard. I'll create like a place for you to upload this assignment as a PDF or as a Word file, and so on. Okay, so let's move to a quick example of how to do this stuff. Here, this is a Google Sheets document. You can create a free Google account if you don't have one. And we can import the data that I uploaded to Blackboard. So I'll just go through this briefly. So we can call this what we want. Let's see. Lab 1 data. So if you go to File, Open, we can go to one of these things that I've already uploaded, or you can just drag a, let's see, I don't know why that's not showing up. There we go. So you can drag your file to upload that we downloaded from Blackboard. And this is the same thing that you would see in Microsoft Excel. And so the first thing we'll do, the first question is about like the relationship between average parent height and the height of their offspring. So here's how you create a scatter plot. So you basically in Sheets, Google Sheets, you have to make sure the columns are next to each other, to my knowledge. If you shift click, you can click multiple cells, right? So if I click on the first cell hold down Shift and click here. You can let go of Shift and scroll down to the bottom. Hold Shift and click here to select the data in the two columns. And then on the toolbar, we can insert a chart. So here we have a chart. It's not really formatted. It doesn't look exactly right, but it brings up this chart editor, and you have different chart types. So you could hypothetically put whatever you want here, but none of these really make sense for us. The scatter plot makes the most sense. So you can change the axes if you have them switched around. This looks good to me. You have to change the, get rid of the title. You have to change the axis labels. But more importantly, click on Customize. And what we want to do is change the scale on the axis. And that's because right now they are unequal. So average parent height ranges from 0 to 200 centimeters. So if you go to vertical axis, you can type in a minimum value. So let's just hypothetically make this 150. Let's make the max value 190. Let's make the horizontal axis have the same range, 150 to 190. You know what? That's actually cutting the data off. So let's go. Let's see. You can just try different values. Okay, 145 seems okay. Let's make that the same for this axis, 145. Oh, there you go. I missed a data point there. So that's really important. Don't cut off your data. I'm going to make this 195 too. Okay, that's better. We can see all the data. The axes are equal. Usually it helps when you're doing this kind of thing to try to make sure the distance between these is square. Right, so now you've got perfect squares or something close to it. That helps us visually reference what we're doing here. Okay. Now, if you click on Series, you can change anything you want. Like you can make the font color gray, or sorry, the symbol color gray. You can change the symbol size. You can change the symbol shape. You do whatever you want. But we want to click trend line. Same thing. We can make this. We can make different kinds of models. Polynomial, logarithmic. Those are different different models that we're not concerned with. We're concerned with a linear trend line, a straight trend line. You can change the line thickness if you want. You can change the opacity, right? How clear it is, so that you can see more points. You can use a custom label if you want, but we want to click on Show R Squared. And so let's see. I don't know what this actually does. Yeah, there we go. So we can show this R squared value, which means that about 15% of, and so we actually have this backwards. Let's go back. It should be like that. Let's see. There we go. Anyway, so you want average parent height on the x-axis, and you want height of the student height on the y-axis. These labels are now wrong, so like we can say offspring height in centimeters, for example, and then average parental height in centimeters. We don't need this. We don't need a title. And there you go. And then you can copy this. Somehow you can save it, or you can take a screenshot, and you can use that to paste into your figures. So you repeat that same kind of process for your different hypotheses and analyses, right? The one looking at latitude and leg length, and the one looking at grip strength and forearm length. And then you'll interpret those results, explain what the R-squared values mean, and so on and so forth. I'm going to upload key terms and concepts for the exam. I'm going to upload the same guide for Microsoft Excel. This is the Google Sheets version. I think this is like the easiest, most straightforward one. Everyone has access to this if you have access to the internet. And so, yeah, hope everybody's doing okay, especially after a stressful weekend and a difficult time. So as I said, please email me if you have any questions. Try to be specific. Try to do your due diligence before you send me an email. And that should be it. I'll talk to you guys next time.